students of IGCSE Combined Science, IGCSE Biology, or perhaps a lover of plant nutrition, welcome to this video. Here I will be teaching you about IGCSE Combined Science B5 topic, plant nutrition. In this video, I will explain both the core and supplement topics 1 to 11 accompanied with a PowerPoint. The core topic includes define photosynthesis as the process by which plants manufacture carbohydrates from raw materials using energy from light, state the equation for photosynthesis, investigate the necessity for chlorophyll, light, and carbon dioxide for photosynthesis using appropriate controls, identify chloroplasts, cuticle, guard cells, and stomata, upper and lower epidermis, palisade, mesophyll, spongy mesophyll, vascular bundles, xylem, and phloem in leaves of a dicotyledonous plant. Describe the importance of nitrate ions for making amino acids and magnesium ions for making chlorophyll. The supplement topics include state the balance equation for photosynthesis, explain that chlorophyll transfers light energy into chemical energy in molecules for synthesis of carbohydrates, outline subsequent use and storage of the carbohydrates made in photosynthesis, and investigate and describe the effect of varying light intensity and temperature on the rate of photosynthesis, e.g. in submerged aquatic plants, describe the significance of the features of a leaf in terms of functions, to include palisade, mesophyll, and the distribution of chloroplasts, photosynthesis, stomata, spongy mesophyll cells, and guard cells, gas exchange, xylem for transport and support, and phloem for transport, and finally, explain the effects of nitrate ion and magnesium ion deficiency on plant growth. By the end of this video, I hope you have grasped enough knowledge from each tightly packed subtopic. From the criteria, I will be splitting the topic into 6 parts: photosynthesis, its carbohydrates, necessities, affecting factors, leaf structure, and minerals. I will be providing the timestamps in the description below. First, we begin with the introductory topic, photosynthesis. As plants are autotrophic, photosynthesis is the process by which plants manufacture carbohydrates from raw materials using energy from light, a metabolic process in photosynthetic organisms requiring light energy to transform raw materials into carbohydrates stored and used for energy. The light energy is first absorbed by the chlorophyll, then converted into carbon dioxide and water into glucose, with oxygen as a byproduct. The word equation for photosynthesis is carbon dioxide plus water into glucose plus oxygen in the presence of light and chlorophyll. Or a balance equation required for the supplement topic, a combination of 6 carbon dioxide molecules and 6 water molecules in the presence of light and chlorophyll produces 1 glucose molecule and 6 oxygen molecules as a byproduct. As you can see, it's already balanced since we started with only 1 of each and gradually multiplying various ions to balance the equation. What is the process of photosynthesis? Photosynthesis in the line required roots absorbing water from the soil and carried to its destination. Carbon dioxide is also absorbed through the stomata. The containment of chloroplasts in palisade mesophills will prove useful in this process due to its possession of chlorophyll. The light energy from the sun is captured by chloroplasts and stored in stacks of the thylakoid membrane. In these membranes, the light energy captured by the chlorophyll is used to split water molecules into hydrogen and oxygen. The oxygen escapes from the leaf, and the hydrogen molecules are added to the carbon dioxide molecules to form sugar. The process produces glucose and oxygen. There may be various terms you are unfamiliar with here: chloroplast, thylakoid, chlorophyll, and palisade mesophyll. I will be explaining them in brief before we delve into it in the following sections. First, mesophyll cells in general are a type of ground tissue found in plants' leaves. They possess large spaces within the leaf that allow carbon dioxide to move freely. Chlorophyll. Chlorophyll is a green pigment found in chloroplasts acting as an energy transducer. Chlorophylls transfer light energy into chemical energy in molecules for the synthesis of carbohydrates. When light falls on a chlorophyll molecule, some of its energy is absorbed and released. The released byproduct assists in the combination of carbon dioxide and water with the help of enzymes in the chloroplasts. And thylakoid. Thylakoid are flattened sacs inside a chloroplast arranged in grana where light reactions of photosynthesis take place. Now, why is photosynthesis important? What is its significance? Photosynthesis is essential for the life of plants since photosynthesis is the production of their food. It obtains fuel and food for the function of plants. It's a pathway for access to solar energy for many living organisms. If photosynthesis ceases to exist, plants will be unable to live up to their function. Due to its significant production of oxygen, then its eradication would lead to little to no oxygen on this planet. Before we delve deeper into the subsequent use of carbohydrates produced in photosynthesis, you should know that we will be talking about the storage and use of glucose, a carbohydrate byproduct of photosynthesis. Glucose has many complex alternate routes. It may go through oxidation, or the loss of electrons, which will then be produced into energy. The energy from glucose may be released within the leaf, and to do so, glucose will be broken down by respiration to release this energy. The next route is it goes through condensation. Condensation happens when a gas turns to liquid. The gas particles will slowly stop moving very rapidly and creating a weak attractive force to form a liquid state. Glucose may condense into 3 different products. The first product is sucrose. Because of glucose's reactive nature, it is transformed into sucrose, a complex sugar, to be transported into other areas of the plant. Sucrose will dissolve in the sap of phloem vessels and may be distributed elsewhere. The sucrose may then be reused as glucose to release energy, stored as starch, or other substances needed for plant growth. The second product is starch. Starch is a polysaccharide made by joins of glucose molecules, and since starch is a large molecule, it is not reactive nor soluble, unlike glucose molecules on its own. Starch is insoluble and it wouldn't affect the osmosis. The osmosis is the movement of a solvent through a semi-permeable membrane into a solution of higher solute concentration that tends to equalize the concentrations of solute on the 2 sides of the membrane. Starch may also be stored in chloroplast as granules. Joints of glucose molecules could contribute to cellulose. Cellulose helps strengthen cell walls. Cell walls need their strength to develop turgor pressure and provide tensile strength and protection against mechanical or osmotic stress. The glucose may undergo reduction into fats and oils. The oils will partake with cell membranes and the fats are used for long-term energy storage in seeds. The next product requires an additional supplement of nitrogen because our 78% nitrogen air is unreactive. Furthermore, the addition of other minerals are depending on the different products. Obtaining magnesium and nitrate ions from the soil, plants may make chlorophyll. Farmers often add their minerals to do so, as deficiency of nitrate may cause weak growth and yellow leaves, while deficiency of magnesium causes yellowing between the vein of the leaves. When glucose combines with nitrate and sulfate from the soil, taken from their root hairs, diffusion, and active transport, it will produce amino acids and protein. And with nitrate and phosphate, glucose can produce nucleic acids that carry information for protein synthesis. And I will further elaborate on their necessity in the following sessions, but for now we will talk about the necessity of light, chlorophyll, and carbon dioxide. Beginning with light. Light is required as a fuel to drive the reaction produced in photosynthesis. For a shred of practical evidence, we may resort to an investigation that describes the effect of light on photosynthesis. Acquire a well taken care of bean or pelargonium plant grown in a pot. Leave it in a cupboard, best if no contact with light, for a minimum of 24 hours so that it may be discharged. Pick one of the leaves to be tested upon regarding the presence of starch to check. And how do we test the presence of starch? First, we drop the leaf into a boiling water bath and leave it be for 30 seconds. This is done to break down the cell walls, and then you turn off the Bunsen flame once finished. You can tell by the softness of the leaf. Gently remove the leaf from the water and place it inside a bottle of ethanol inside the said water bath. This is done until the chlorophyll is completely out of the leaf, known by the pigmentation of the alcohol. Due to the leaf's brittle state, we must now put it back into the boiling water to soften it. Now test the leaf for starch using an iodine solution by seeing the pigmentation. Following the discharging, partially cover another leaf with aluminum foil or black paper with the edges fastened using clips if necessary. Leave the leaf in a place rich of solar energy. The final step is to test the leaf for starch using an iodine solution. The area of the leaf that was covered with aluminum foil will remain orange-brown as it did not receive any sunlight and could not photosynthesize, while the area exposed to sunlight will turn blue-black with the presence of starch. We further continue with the necessity of chlorophyll. Chlorophyll absorbs the light energy necessary for photosynthesis. Without the chlorophyll, there will be little to no light acquired for photosynthesis, losing the fuel to drive the reaction produced. To further prove this, we may resort to the previous investigation. Destarch a plant with variegated leaves. Leave it in a cupboard best of no contact with light for a minimum of 24 hours so that it may be destarched. Test one of the leaves for starch by dropping the leaf into a boiling water bath. Leave it be for 30 seconds. This is done to break down the cell walls. Turn off the Bunsen flame once finished. You can tell by the softness of the leaf. Gently remove the leaf from the water and place it inside a bottle of ethanol inside the said water bath. This is done until the chlorophyll is completely out of the leaf, known by the pigmentation of the alcohol. Due to the leaf's brittle state, we must now put it back into the boiling water to soften it. Once finished, place the leaf on a white tile and cover it with iodine solution. With the presence of the starch, the pigmentation turns blue-black and successfully photosynthesized. Without the starch, the color will instead be brown and failed to photosynthesize. With eradicating the chlorophyll, we can conclude the reaction to be brown, thus a failure in conducting photosynthesis. The investigation proves the necessity of chlorophyll for the production of starch and obtaining light. The following topic will be the necessity of carbon dioxide for photosynthesis. Carbon dioxide is an inorganic compound essential for photosynthesis because of its ability to convert into glucose. Once again, we may conduct an investigation. First, we must destarch a plant. Leave it in a cupboard, best of no contact with light, for a minimum of 24 hours so that it may be destarched. Test one of its leaves for the presence of starch. Drop the leaf into a boiling water bath and leave it for 30 seconds. This is done to break down the cell walls. Turn off the Bunsen flame once finished. You can tell by the softness of the leaf. Gently remove the leaf from the water and place it into a bottle of ethanol inside the said water bath. This is done until the chlorophyll is completely out of the leaf, known by the pigmentation of the alcohol. Due to the leaf's brittle state, we must now put it back into the boiling water to soften it. Test the presence of the starch using iodine solution. Tie a clear bag with sodium hydroxide surrounding half of a selected leaf. This will act as the carbon dioxide absorbent surrounding the leaf, providing the leaf inside with no carbon dioxide at all. Tie a clear bag with water surrounding another leaf. This will act as the control as the leaf will still have access to carbon dioxide. Place the plant in an area rich in bright light. Pick both leaves and test it for the presence of starch. Place the leaves on a separate white tile and cover them with iodine solution. With the presence of starch, the pigmentation turns into blue-black, while with no presence of starch, it will turn brown and fail to photosynthesize. The leaf from the bag containing sodium hydroxide will remain orange-brown as it could not photosynthesize due to the lack of carbon dioxide. The leaf from the control bag containing water should turn blue-black as it had all the necessary requirements for the photosynthesis. We now carry on to the topic of factors affecting the rate of photosynthesis. Before we delve deeper, we must understand the basic investigation of the rate using types of pondweed, for example, Alodia or Cambula. First, place the pondweed in a large beaker filled with a combination of water and sodium hydrogen carbonate, which is the controlled variable. Place an inverted funnel to cover the photosynthesizing aquatic plant, another controlled variable, and cover the funnel with an inverted boiling tube to catch the bubbles. Use a thermometer to monitor the temperature since it is another controlled variable. As photosynthesis occurs, oxygen gas produced is released as bubbles. Thus, we may investigate the rate of photosynthesis by measuring the bubbles produced over a minute. This investigation will be the basis of the later investigations we may conduct. The first factor affecting the rate of photosynthesis is the intensity of light. The intensity of light is directly proportional to the rate of photosynthesis. If the amount of light is insufficient, the rate of photosynthesis will slow down despite the presence of carbon and water. However, the high light intensity may lead to heating the temperature, and the rate will instead be constant because of the interruption of other factors. To further investigate, we may conduct this experiment measuring the effect of light intensity on photosynthesis. Place the pondweed in a large beaker filled with a combination of water and sodium hydrogen carbonate, which will act as our control. Place an inverted funnel to cover the photosynthesizing aquatic plant, another control, and cover the plant with an inverted boiling tube to catch the bubbles. Use a thermometer to monitor the temperature since it is a controlled variable as well. Place a glass tank filled with water not too far from the large beaker, and the glass tank will absorb the heat. Beside it, place a lamp. The factor being investigated is the distance of the lamp, so we must use a ruler for more accurate data. Move the lamp different distances away from the beaker containing the pondweed. Investigate the rate of photosynthesis by measuring the bubbles produced over a minute. The rate will slow down the farther the distance of the light will be, and that is our final conclusion. The second factor affecting the rate of photosynthesis is the temperature while conducting photosynthesis. This is because the temperature has a direct effect on the enzymes of the plant, enzymes necessary to carry out photosynthesis. The temperature for photosynthesis is usually 25 degrees Celsius due to the required enzymes having an optimum temperature. At low temperatures, enzymes have little kinetic energy, which results in a slower rate of photosynthesis. And at higher temperatures as well, enzymes may denature, causing a slower rate as well. Once again, we may investigate with a practical approach. Place the pondweed in a large beaker filled with a combination of water and sodium hydrocarbonate, which is our control, and place the beaker on top of a hot plate. Place an inverted funnel to cover the photosynthesizing aquatic plant, another control, and cover the funnel with an inverted boiling tube to catch the bubbles. Use a thermometer to monitor the substance since we will be measuring the variable. Put a lamp at a constant distance due to light intensity being controlled. Change the temperature of the water using the hot plate. Investigate the rate of photosynthesis by measuring the bubbles produced over a minute. From the investigation, we may conclude a required fixed temperature that makes photosynthesis more efficient. And we may also conclude the effect of temperatures on photosynthesis. The final factor we'll be talking about is carbon dioxide. Carbon dioxide deficiency will lead to a terrible photosynthesis. Carbon dioxide is directly proportional to the rate of photosynthesis, so if it increases, so will the rate of photosynthesis. This trend will continue until some other factor required for photosynthesis prevents the rate from increasing further because it is now in short supply, and we may investigate as such. Place the pondweed in a large beaker filled with a combination of water and 1% of sodium hydrocarbonate solution. Place an inverted funnel to cover the photosynthesizing aquatic plant, which is our control, and cover the funnel with an inverted boiling tube to catch the bubbles. Use a thermometer to monitor the temperature since it is a controlled variable. Place a glass tank filled with water not too far from the large beaker, and beside it would be a lamp. These should be at a fixed distance from the beaker so it is controlled. The factor being investigated is the percentage of sodium carbonate in the water. You may investigate the rate after constantly adding the substance and comparing them. After talking about photosynthesis, its carbohydrates, necessities, and affecting factors, now we will talk about the parties who contribute and their involvement. We will discuss leaf structure and its significance. Here is a glance at the overall structure as we will slowly unravel component after component. The first component we will talk about is chloroplast, the place where photosynthesis occurs. Chloroplast contains chlorophyll, a green pigment found in chloroplast acting as an energy transducer. Chlorophylls transfer light energy into chemical energy in molecules for the synthesis of carbohydrates. Cuticle. Cuticle is a protective clear wax-like layer covering epidermal tissues of a leaf, providing water resistance properties and reduces water loss, thus preventing water from evaporating. A guard cell absorbs and releases water in order to open and close the stomata, which allows the diffusion of carbon dioxide in and oxygen out. Guard cells also contain chloroplasts, unlike other cells in the epidermis. Stomata or stoma, where the water evaporation and additionally gas exchange of the plant occurs. Opens during the day and closes during the night. Commonly found on the underside of leaves to reduce water loss. The reduction of water loss, however, additionally reduces the diffusion of carbon dioxide as well, slowing the rate of photosynthesis. Plants in places of higher temperature tend to have fewer stomata. Following the stomata, we have the upper and lower epidermis, a protective barrier preventing pathogens from causing harm. Epidermis cells are transparent to light let pass through and do not contain chloroplasts. The lower epidermis additionally contains guard cells and stomata. Palisade mesophyll, cells column-shaped that are tightly packed with chloroplasts. The function of a palisade is to increase the absorbance of light efficiently. It is strategically placed on top of the leaf, rich in sunlight. Its containment of chloroplast creates an adaptive nature. An additional responsibility for palisade mesophyll is its distribution of chloroplasts. Light absorption happens in the palisade mesophyll tissue of the leaf, tightly packed with chloroplasts. Its close arrangement results in a more efficient absorption of light energy. Spongy mesophyll, placed in the lower layer of the leaf. Spongy mesophyll cells possess a large surface area and internal air spaces, allowing the rapid diffusion of carbon dioxide, water, and oxygen if the stomata is opened. Gas exchange in plants happen in the spongy mesophyll tissue. While plants are performing photosynthesis, the spongy mesophyll allows the diffusion of carbon dioxide in and oxygen out. Gas exchange is assisted with stomata and guard cells as well, as mentioned before. The reason why gas exchange is important is due to the necessity of energy. All living organisms require energy to carry out their daily activities, and they obtain them through cell respiration, the oxidation of food. Cellular respiration demands oxygen and its production of carbon dioxide. Thus, to acquire energy, all living organisms need to obtain oxygen and expel waste carbon dioxide. Last but not least, we have vascular bundles. The vascular bundles or veins form the transport system of the plant. It contains xylem and phloem, which transports numerous substances into and out of the leaf. Next, made out of vascular sheeted bundles of cells. Xylem and phloem are tubular cell networks allowing transportation of various substances. Phloem is used as a transport medium for sucrose and amino acids around the plant. Phloems are made of living cells and may be adapted with no nucleus. The translocation of substances goes both up and down the stem. The phloem is needed both directly and non-directly for seed development, storage organs such as bulbs and tubers, and growth. Meanwhile, the xylem is a transportation medium for waters and minerals, sending it to the mesophyll cells to photosynthesize and stomata to transpire. Xylems are made up of hollow-out dead cells with the ends removed for transportation purposes, having thick cell walls for structural strength, but thin enough to allow capillary action. At the moment, we have reached our final topic regarding plant nutrition. Here we will discuss the importance of nitrate and magnesium ions along with the consequences of their deficiency. Nitrate ions are used to build amino acids, thus proteins and enzymes. Its deficiency would affect the growth and or repairment of a plant. Alternatively, magnesium ions assist in the making of chlorophyll. If a plant lacks magnesium ions, it will lead to lack of chlorophyll. Chlorophyll plays a key role in the photosynthesis. Without it, plants will not be able to form sugars. Therefore, the plants won't have a sufficient enough energy source for growth and repairment. With this reason, farmers often add their own minerals in order to enhance their plant, as deficiency of nitrate may cause weak growth and yellow leaves, while deficiency of magnesium causes yellowing between the veins of the leaves. We have reached the end of B5. Thank you very much for your avid attention. I'd like to remind you that by the end of this video, we have discussed subtopics 1 to 11, and thus you should be able to understand and answer these questions on your own. I suggest Quizlets for active recall and memory retention, or perhaps if the information came through one ear and out of the other, repeat this video or feel free to check my references. If you're super sure, I suggest doing past paper questions of B5 to test your knowledge, which I will provide in the description below. Once again, thank you very much. Good luck for your exams.